Good afternoon, everyone. My name is Mark Levenson. Today I'm going to be talking about statistical principles for clinical development. There are two learning objectives to my presentation. The first is to understand basic statistical concepts such as bias, variability, and multiplicity. And the second is to understand the role of statistical concepts in good study design, conduct, and analysis. So basically, the first learning objective will lead into the second. Here's my outline. First, I'm going to give a quick review of good study principles. Dr. Smith has already presented on much of this. And then the heart of my talk is I'm going to go into statistical concepts and the implications on study elements. And this will start with a discussion of the distinction between bias and variability and the implications of each. And then I'm going to drill down into variability and then bias. And finally, I'll provide some concluding statistical principles. Okay, just to get some terminology down that I'll be using throughout my presentations, I'd like to provide some terms for the stages of a study. The stages start at the design stage. This is the conception, planning, and specification of the study. Typically, this will result in the protocol and statistical analysis plan, or SAP. The second stage is the conduct. This is the running of the study. This includes recruitment, screening, intervention, concomitant care, outcome ascertainment, and safety and study monitoring. The next stage is the analysis of the study. This is the number crunching, going from the raw data to the final results. Typically, this is where people believe the statistician comes into play. But I argue that the statistician has a role in all the stages of a study. Finally, there's reporting. The reporting of the study should basically align with the protocol and statistical analysis plan. I won't go into reporting much today, but there are guidelines on this. A common theme or message in my presentation is that design and conduct are more important than analysis. In other words, analysis cannot make up for poor design and conduct. One should focus on good design and conduct, and the analysis will be straightforward. And statistical concepts motivate good design and conduct, and that's really the point of my talk, is to show where statistical concepts can improve design and conduct and result in overall a better study. So what are the goals of study design, conduct, and analysis? The first goal is very clear. You're trying to address a specific clinical question. That's why you're running the study. I'm going to present two additional goals that come into play as you choose design and conduct decisions. The first one is to minimize bias and minimize variability. I'm going to spend a lot of time on what that means, both for bias and variability. And finally, you have to be able to ethically, safely, and feasibly conduct the study. You can't do a study that's not ethical for patients, not safe for patients, or it's just not feasible. Now, the problem is there's some conflict among these goals. There's almost inevitably some conflict among these goals. And let me give you an example. Okay, here's an example of balancing these three goals. And this is the concept of enrichment versus generalizability. So the clinical question we're interested in is a drug effect on cardiovascular events for all patients with type 2 diabetes. And the emphasis here is on all patients. This is an event trial, so we're trying to measure cardiovascular events. Event trials usually require a certain number of events to get the necessary statistical precision. You can get more events for the same number of patients if you study patients with additional CV risk factors. For example, patients that may have had a prior heart attack. So for the same number of patients, if your patients are at greater risk, you're going to be able to run a smaller trial. However, you're not really answering the first clinical question that I stated because you're interested in all patients with type 2 diabetes. So here we see that there's a balance between feasibility and answering the clinical question. Okay, that was my quick review of good study principles. I'll return to many of these principles throughout the talk, and now I'm going to get into the heart of the presentation, and that is the statistical concepts and their implications on study elements. And first, I'm going to define bias and variability and the important distinction between the two. Okay, here we have four bullseyes. Let's start in the upper left, low variability and low bias. The dots are intended to represent your data points, and the center of the bullseye is intended to represent the truth, the answer you're trying to seek. So in this bullseye on the upper left, we see how data points are packed closely together. That's the low variability, and they're actually centered on the center of the bullseye, and that's the low bias. Moving to the upper right, we see that the data points are spread apart, and that's the high variability, but they're generally centered on the average around the center of the bullseye, and that's the low bias. Moving to the lower left, the data points here are clumped together with low variability, but they're far from the center of the bullseye, so this represents high bias. And finally, the combination of the two, in the lower right, we have the data points spread apart and not really centered around the middle of the bullseye, and that's high variability and high bias. So those basically informally define these two terms. Now the problem is you don't get to see the bullseyes. So say these were your data points, and I ask you the question, do these data have low bias? Well, I mean, let's say, are we able to even answer, do they have low variability? Well, that's a relative term, but using kind of the scale we had on the last slide, let's say these data points have low variability. But because we don't have the bullseye here, we don't know the true answer, because that's why we're running the clinical study. We don't know if these data points are centered on the truth or centered around some value away from the truth. So we don't know about the bias in this case. I hope those few slides demonstrated a few things. First of all, I hope they demonstrated what bias and variability ultimately are. But there are some other important concepts I hope they demonstrated. First, that we see and can measure variability. We can see how the data points are spread apart from each other. We can measure that using such measures as standard deviation and ranges. We do not see bias. We don't know what the true answer is, or not we wouldn't be running the study. Bias is mainly addressed in the design and conduct stages. And I hope the statistical principles I bring out through this talk will help motivate good design and conduct. Now we have a definition of bias and variability, and I'm going to be drilling down into each of them. First, variability and associated statistics and concepts related to that. So variability and sample size. So sample size, of course, is the number of people in the study. I'm going to ask you a question. What is a better estimate of the average age of this session's attendees? Now, there may be over 1,000 attendees in this class, and we would like the average of those 1,000 values, but we're going to take a sample of the attendees, as is often done. So the first possibility is we pick a random sample of 5 attendees and calculate their average age. The second choice is to pick a random sample of 50 attendees and calculate their average age. Now, which of this is going to give you a better average age estimate of the overall session attendees? Clearly B will, because more people are involved in this sample. Some people will have high values relative to the overall mean, some will have low, and the more you pick in your sample, the more there's going to be cancellation, and there's going to be less variability in these larger sample sizes. Now I'm going to introduce a little notation to further explore variability and sample size. Let's call x hat 5 the mean of 5 people, and x hat 50 the mean of 50 people. We intuitively saw on the last slide that x hat 50 has less variability than X hat 5. Let's look at variability and sample size a little more mathematically. The standard error of X hat 50 is a measure of the variation of X hat 50. And the standard error is defined as the standard deviation of X. Those are the individual values of X, the individual ages in the population, divided by the square root of n, the sample size. So as you can see from this formula, as the sample size n gets larger, the denominator gets larger and the ratio gets smaller. So for larger sample sizes, the standard error decreases. Okay, we're going to get into a little bit more math. I'm going to introduce the Z-statistic. The Z-statistic is the effect estimate. So that's basically what you're trying to estimate, like the effect of a drug, over the standard error of the effect estimate. So this is an estimate over a standard error. It's basically a signal to noise quantity. Just to make this a little concrete, I'm going to assume the effect estimate is 0.5 meters. Now I'm going to be using kind of a running example through a few slides that the endpoint we're interested in is how far you can walk in 6 minutes. And what we've observed in this study is that there's a 0.5 meter greater distance that people on drug can walk than on control. That's a small amount, but that won't be important right now. As the sample size increases, we saw on the last slide that the standard error goes down. And as the standard error goes down, because it's the denominator in this quantity, the Z-statistic goes up. And what I won't really show is as the Z-statistic goes up, the p-value goes down. What that means is it's more significant. The same result, the 0.5 meters, if you observe that in a larger study, that will give you a more significant result. Let's continue this. Typically, if there is no drug effect, then the probability of seeing the absolute value of the Z-statistic greater than 2 is 0.05. So 0.05 is 1 in 20. And absolute value means if we see -3 or +3, they're both greater than 2. If we observe an absolute value of the Z-statistic greater than 2, then we say the finding was significant at the two-sided 0.05 level. So what this says is if there was no drug effect and we observed, say, a Z-value of 3, that was a pretty unlikely event, certainly less than 1 in 20. And that gives us doubt that there really is no drug effect, and maybe the assumption of no drug effect is wrong. So we call that statistically significant. It's a tricky concept, but that's basically what it is. Now let's return to the effect of sample size. For a given real effect, say 0.5 meters, the probability of a significant effect increases with sample size. So we saw that on the last slide. If the effect is 0.5 from a larger study with more sample size, it will have a smaller p-value, more significant than the same effect from a small study. So when we say a larger study has more power than a smaller study, because the probability of seeing a statistical significant result for the same effect size is larger. So to say that again, larger sample sizes have higher power. Now I will move on to the concept of multiplicity, also known as multiple bites from the apple. Suppose I told you a study found a drug had a statistically significant effect for females aged 51 to 60 years old. Well, that sounds like an important finding. Now suppose I told you the study actually studied people from all ages, from the first year of life to people over 90, and both males and females. And they calculated the Z-statistic for every combination. So each of these cells gives you a chance of the Z-statistic being greater than 2, even if there's no drug effect. So perhaps it's not surprising that some were greater than 2. In fact, greater than 2, even if there's no drug effect, occurs 1 in 20 in a 1 in 20 chance. So here it's not at all surprising that if we looked at enough of these different combinations, one of them would be greater than 2. But it doesn't, I think, call into question that there's no drug effect. It very likely could be no drug effect, and you would see a result like this. So multiplicity, even if a drug has no effect, if you conduct enough statistical tests without proper adjustment, you'll find some results statistically significant. This is known as multiplicity. And what the implication is here, that although they're nominally statistically significant, they're not really statistically significant. They're not unusual findings. So multiplicity can show up with multiple subgroups. So we saw age and sex in a few slides ago, multiple endpoints or analyses. So subgroups might be effect on males, effect on females, effects on people over 65. Endpoints might be effect on blood pressure, effect on life expectancy, effect on happiness. And finally, if you even run the same different statistical analyses on the same subgroups and same endpoints, you may get different results. So for example, if your primary analysis is some sort of multiple regression, but use different covariates, you actually may get different results. So multiplicity can come up in a whole variety of ways. There are solutions to the multiplicity problem, and that the primary solution is pre-specification. You tell the world ahead of time what you will primarily look at. So in that study a few slides ago, if the study had primarily ahead of time decided that they're most interested in females of a certain age, and they found a statistically significant result in that subgroup, that would be an important finding. At least that would be a statistically significant finding. So this avoids what's known as fishing for p-values, looking at multiple subgroups or endpoints until you find something that has an interesting looking p-value. So the way you tell the world ahead of time is through protocols and statistical analysis plans. So those should be completed before the protocol should be completed before the start of the study, and the statistical analysis plan is usually completed before unblinding of any data. Now, there are statistical methods that can handle multiple outcomes or multiple subgroups. I mean, suppose you genuinely are interested in 2 different endpoints, like blood pressure and survival. There are statistical methods that can properly adjust just for these multiple comparisons and this multiplicity. But I certainly won't go into them today, but the important thing is they need to be pre-specified too. We spent a little time on variability, and now I'm going to move on to bias. So in bias, I'll cover various types of bias and say something about sensitivity analyses. This section will be longer than the variability section, but more intuitive, I hope. Okay, we'll go back to this formula for a Z-statistic. There's nothing new here. We have a Z-statistic, that's the effect estimate over the standard error of the effect estimate. And for a given effect size, say 0.5 meters, if the sample size goes up, the standard error goes down, the Z-statistics goes up, and the p-value goes down, meaning it's more significant. Let's look at a bias example. Here we have an unblinded trial, and the endpoint is effort dependent. I'm going to use the 6-minute walk example again, how far you can walk in 6 minutes. Because patients know what drug they're on, whether they're on the experimental drug or the control, patients on the experimental drug may try harder. This may not be a conscious bias. This may be more of an expectation bias. They think they're on some novel therapy, so they're going to be doing better. Let's say on the average that they do 0.5 meters better. Some may do more than 0.5 meters, some may do less, but on the average, this expectation bias makes people on the experimental drug do 0.5 meters longer in a 6-minute walk. Let's assume there's actually no drug effect here. Now, the important point here is this 0.5 meter bias will not likely be affected by sample size. Everybody will have, on the average, this 0.5 meter bias. It's going to appear no matter what sample size you have. So p-values will get smaller for larger sample sizes, even though the drug is not affected. We've learned something about addressing bias. Sample size does not address bias. A larger sample size will not get rid of bias in most cases. What you need is design and conduct. You use design and conduct to address bias. So some of the design methods are randomization, blinding, good outcome ascertainment. What I'd like to say is analysis cannot rescue poor design and conduct. Analysis can address some bias and explore sensitivities to bias, which I'll talk about at the end of the talk. So in order to help identify biases, it's useful to categorize them. So I'm going to categorize 3 types of biases, and it's not an exhaustive list, but it accounts for much of the bias you may see. The first is confounding. This has to do with baseline differences between treatment groups, and this is addressed by randomization. I'll go more into this. The second type of bias to have your eye out for is measurement bias, or the endpoint ascertainment. This is typically addressed by blinding and reliable and valid measures. I really won't be saying any more about this in today's talk. And finally, there's selection bias. This includes both baseline and post-baseline population issues, which I'll have a bunch to say about in a few slides. Confounding. So without randomization, there may be systematic differences, bias, when comparing people getting drug A and people getting drug B. This is known as confounding. Let's take an example of this. Suppose drug A may be given to older, sicker patients. Perhaps drug A has less side effects, and older and sicker patients can tolerate it better. So even if there was no difference between the effects of drug A and drug B, the comparisons may show that drug A has worse outcomes because drug A is made up of sicker patients. Again, this is known as confounding. Now, I already mentioned, randomization addresses confounding because it makes the 2 comparator groups comparable at baseline. Many of the studies you see in the paper are not based on randomized studies. So if you ever hear anything about coffee or red wine or chocolate, chances are it was not based on a randomized study, and I would take the results with a grain of salt. Now let's move on to selection bias, and I've broken that up into 2 types. So the first type is baseline study population is not representative of the intended population. This is a relatively easy concept to understand. So let's take an example. So suppose in your study there are a few people with comorbidities. So this will affect the generalizability or the external validity of your study, that what you study may not represent the larger population, in this example, people with comorbidities. The second type of selection bias is that the analysis population is based on post-baseline information. This is harder to understand, and I'm going to spend some time on this. So some quick examples that we'll dive deeper into are analyzing adherers only, analyzing those with no missing data, and analyzing those with an initial response to treatment. You can see that the population you're analyzing is all based on post-baseline information. What this does is it affects the causal relationships, the actual relationships you're trying to study, the drug effects. And this is actually an effect on internal validity. Even the relationships you estimate within your study are wrong, let alone that they don't generalize to a larger population. So here's a classic example of this from an old study, the Coronary Drug Project, that's often used to illustrate this point. So how adherence itself may be related to the outcome. So here the drug clofibrate is being compared to placebo for a mortality outcome. So on the bottom here, we have the percent of people, the percent of mortalities in the clofibrate group, which is about 18%, and the percent in the placebo group, which is about 19%. So there's not much difference here. It doesn't look like clofibrate has much effect on this population on mortality. But there's actually more to this table than I showed you initially. This table also breaks down the results by your adherence, whether you have less than 80% adherence or greater than 80% adherence. This is basically how much of the drug you took compared to what you were supposed to take. And what we see for the ones with higher adherence, those with greater than 80%, the mortality is down to 15%. And those with less than 80%, the mortality is close to 25%. So if you compare that 15% of the adherers, the people who actually took the drug a reasonable amount of time, to the placebo amount, it looks like the drug has an effect. But the thing to be revealed here is the placebo group has the same pattern. So the people who took placebo more than 80% have lower mortality than the people who took placebo less than 80%. Now, obviously, taking placebo more is not going to affect your mortality. What's really being demonstrated here is the people who have a higher adherence are different from the people who have lower adherence. They may have more attention to their own health, or there are any other number of differences between the two groups. And this difference, based on a post-baseline factor, can introduce bias, which doesn't show up if you do not divide by adherence, but when you start looking at adherence, you see that there are differences. So let's talk about adherence and missing data and biases. Patients who complete therapy may be different from those who stop therapy. I think we could think of several reasons for that. The people who complete therapy, again, may be more interested in their health. They may be less sick. They may have less side effects. So there's a difference. So treated group patients who complete therapy may be different from control patients who complete therapy. Now this is very subtle, and you might not be able to pick it up in the time I have on this slide, but the first bullet talks about if those who complete therapy are different from those who stop therapy. But this difference actually may be different between the two treatment groups. So if the first bullet was the only problem, then maybe you would just look at people who adhere to the drug. Like in the last example, those with greater than 80% adherence. But the reason people don't adhere may be different for patients on treatment versus those on placebo. For example, maybe this drug has a lot of adverse events associated with it. So the people prone to adverse events stop treatment. But on the placebo, those who stop it may be stopping it. They're not stopping it for adverse events because they're only taking placebo. They're stopping it for other reasons. So this is a rather subtle point, but it explains why we just can't look at adherers. Analyzing only patients who complete therapy, or what we call complete data, will likely produce biased results. So that's the kind of conclusion of all this. So let's talk about the different types of adherence and missing data, because this has implications on how you address them, in the design and analysis. So one type is patients may stop treatment. They may stop treatment, but you may actually be able to follow them up after they stop treatment. So stopping treatment is not the same thing as leaving the study. Leaving the study is the patient is not agreeing to have any more follow-up, or has just been lost to follow-up perhaps. So a patient can stop treatment and still be followed up. So I'd like to distinguish those two situations. And a third situation is, patients may miss a single study visit. That visit might have involved a particular treatment or a particular assessment, perhaps the endpoint assessment. So you may be missing some of your endpoints. So these are three different types, and it may be important to think of them separately and how you might deal with them. But they're all called intercurrent events, and they should be handled appropriately and not often the same way. So what causes, I've already talked a little bit about this, but what are some of the causes of lack of adherence or missing data? Well, the first thing might be random. So it's not related to anything about the patient's characteristics or the patient outcome. So if this was the only problem, then we could just ignore it, basically. The patients that have the information are just like the patients who don't have the information. It's all random. We could just use the patients where we have the information. We would lose sample size, but we wouldn't create any bias. Another possibility maybe is the frailty of the patient, like sicker patients drop out. And again, maybe it's lack of efficacy. Maybe the patients who are perceiving not getting any benefit from the drug are more likely to drop out. And also I've mentioned side effects. Maybe certain side effects makes patients drop out. Again, it's important to think about this as you design your study and as you come up with analyses and eventually sensitivity analyses. So just a word on adherence. I don't want to make it imply that adherence is forcing the patient to do something. This is not ethical. The protocol should allow for the best interest of the patient. So we're not trying to force adherence. We're trying, the study will represent what happens. We may encourage adherence, through education efforts or whatever, but we're not trying to force the patient to do anything that's not in their best interest or not that they're willing to do. So adherence is basically following the protocol, which should allow for the best interest of the patients, even if that means stopping therapy. Okay, so I've already talked about some reasons for missing data, some reasons for lack of adherence. So what are some of the solutions for that? The primary way we analyze efficacy trials typically is intent to treat, which is currently also known as treatment policy. So the first step is we want to get follow-up data regardless of treatment adherence. So even if someone stops treatment, we still like to get the follow-up data. So as I said earlier, stopping treatment is not the same as stopping the study or dropping out of the study. The patient may, once they stop treatment, may not choose to be in the study anymore, and that's their right and choice. But to the extent that patients are willing to continue to be follow-up, even if they're not getting treatment, has an advantage, because we can see what happens to these patients. So we analyze patients according to their random assignment, regardless of adherence and missing data. So this answers the question, what is the effect of being assigned therapy? So you may stop therapy, you may start another therapy, but it does answer the question, if you're randomized to this therapy, what is the effect? And importantly, it maintains the randomization, because you're still comparing people randomized to one group versus the other group. Now, there are problems with the treatment policy or the intent to treat approach. Patients may stop therapy, they may cross over to the other group therapy, or they may receive rescue therapy. So stopping therapy is pretty obvious, we already talked about it. In some studies, patients on the placebo group may actually start taking the investigative agent, and that could be because it's available through some other means or as part of the protocol. Or some protocol often will allow for rescue therapy. If a patient worsens, they don't allow it just to continue to worsen. Some sort of approved rescue therapy is used for that patient. So this creates sort of complications in interpreting this intent to treat, because these things may happen to a patient, and may affect your view of like the efficacy of the drug, but are not accounted for in this ITT approach. The ITT approach is basically what it means to, what the effect is to be assigned therapy, not what actually happens to you. And unfortunately, you still may have missing data. So ideally, if a patient stops therapy, we would recommend that you continue to collect data as they would if they were still on therapy, or maybe some abbreviated version, or whatever it takes to get the endpoint you need. But a patient may choose not to continue to be followed up, or they may be lost to follow-up. So you still may have missing data. So if to do the intent to treat, you need data on all the individuals. So this is sort of a limitation. So you'll have to find some way of handling this missing data. One approach, not so much for the missing data, but for some of the issues in rescue therapy and stopping, is to use a composite approach. So a composite approach would treat stopping therapy, changing therapy, or the use of rescue therapy as a failure in a composite endpoint. So suppose your endpoint was some sort of clinical response, a success or a failure. So the composite would count as a failure not only whatever is being measured, but any of these events like stopping, changing, or rescue would be considered a failure as well. So that's one way to kind of address some of these issues of, that placebo patients may have got rescue therapy, so they're now going to look better. But if you consider that a failure in this composite approach, the ITT approach still works and it still maintains randomization. Again, it doesn't address the missing data, but I'll get to that in a bit. But in general, the approach should be tailored to the specific clinical question you have. Now I'm going to say something about missing data, but mainly use it as an example of sensitivity analysis. Sensitivity analyses involve varying assumptions to explore the potential biases. And the setting we're going to use is the drug effect for patients with missing data is not the same as the drug effect for patients with complete data. I've already said that we don't see bias in the data, but we can use sensitivity analyses to explore assumptions on the bias. This is a rather complicated figure, so I'll spend some time on it. It comes from a study where the primary result was statistically significant, but like all studies, there were missing data. So the green areas in this plot are under certain assumptions where the drug is still statistically significant, and the red areas are where the drug is no longer statistically significant, and there potentially could have been yellow areas where the placebo was actually statistically significant, but that didn't occur. So what we're seeing here is various assumptions of the missing data. The x-axis is assumptions on the estimated mean of the endpoint for those with missing data for the treated group, and likewise the y-axis, the vertical axis, is assumptions for the estimated mean of the control group with missing data. So most of the patients are not missing data, so that they're still in this analysis. But for the patients that are missing data, we make assumptions of what their mean value would be. And we see different regions where the study is still statistically significant. So this involves clinical interpretation, like what would be reasonable to assume for these missing data. If you can make somewhat extreme assumptions and still see a statistically significant result, then maybe the bias from the missing data is not a big deal. So this involves, again, clinical judgment, but allows you to explore different assumptions for the missing data and maybe see how strong the primary result is. This brings me to concluding statistical principles. In summary, careful design and conduct are needed for reliable results. Randomization addresses baseline biases. Blinding, good adherence, good follow-up, and good endpoint ascertainment addresses post-baseline biases. Pre-specification is important to address multiplicity. Here are my take-home messages, returning to the theme of the beginning of the talk. address the clinical question, minimize bias and minimize variability, and careful design conduct will promote valid and reliable results. Challenge question number 1. Which of the following does not reduce bias? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. The answer is a larger sample size. If we go back to that example in the unblinded trial with the effort-dependent endpoint of the 6-minute walk and the belief that patients on the experimental treatment may try a little bit harder, on the average, every patient is going to have that 0.5 meters extra effect on the experimental arm, and that bias is not going to go away with larger sample size. Randomization clearly addresses confounding, and blinding clearly addresses endpoint ascertainment, so they do address bias. And I'll leave pre-specification for a moment. The second challenge question is, which of the following addresses multiplicity? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. Here the answer is pre-specification in the protocol and statistical analysis plan. We talked about multiplicity, that if you look at enough things, you're going to see something statistically significant. So pre-specifying what you're primarily interested in will avoid that issue. So with say thank you, and I'm looking forward to answering questions in the question and answer period.